Hi, in this short presentation, we will look at the role of intercellular chemical messengers in homeostasis. Essential to reflexes and local homeostatic responses, and therefore to homeostasis, is the ability of cells to communicate with one another. In this way, cells in the brain, for example, can be made aware of the status of activities of structures outside the brain, such as heart, and help regulate those activities to meet new homeostatic challenges. In the majority of cases, intercellular communication is performed by chemical messengers. There are four categories of such messengers: hormones, neurotransmitters, paracrine, and autocrine substances. This picture was from previous edition of Wonder. From this picture in the new edition, a part is removed. This part is removed. Here you see the hormone secreting gland. So this is an endocrine gland, releases the chemicals called hormones into the blood circulation, and these hormones affect the target cells, which have receptors to these hormones. Here you see a nerve cell releases neurotransmitter. These neurotransmitters will bind to the receptors on the opposite side of the synapses. It may be a neuron or a muscle cell sometimes. So it is an effector cell then. So it can either depolarize the opposite neuron or effector cells, causing action potential formation or contraction in the muscles, for example. And here you see that the hormone is synthesized in the nervous system. This part is removed in the new edition of Wonder. I think that this is important. That's why I kept this picture also in this presentation. There are nerve cells in the nuclei in hypothalamus, for example, and these neurons are stored in posterior hypophysis and released into the blood circulation. So these hormones are oxytocin and vasopressin. So oxytocin is a hormone synthesized in the nervous system, released into the posterior hypophysis, and from the posterior hypophysis it is released into the blood circulation. For example, when the baby sucks her mother's breast, and via reflex mechanism, oxytocin will be released from the posterior hypophysis to contract myoepithelial cells around the mammary cells, so that milk can easily reach the mouth of the baby. Okay. This oxytocin is also important during delivery of the baby. When baby's head presses on the cervix uteri, and then again it stimulates the reflex mechanism, increasing the oxytocin release from the posterior hypophysis, and oxytocin increases the contractions of the uterine muscle so that the baby can be delivered. Vasopressin is another hormone synthesized in hypothalamic nuclei and transferred to the posterior hypophysis to be stored there. When it is needed, it will be released into the blood circulation. When blood volume goes down or when blood osmolarity increases, then it will stimulate the vasopressin or antidiuretic hormone secretion from the posterior hypophysis, and then it will increase the water reabsorption in the collecting ducts in the kidney, so that increasing the blood volume and diluting the osmolarity in the blood and extracellular fluid. Okay, here you see the paracrine and autocrine secretions in the below, and the same below. This is the same picture now updated in updated Wonders Human Physiology textbook. This is hormone secretion, and this is neurotransmitter release. As you see, hormone secretion from the neurons is removed from the picture. I think it is important, and it would be better if it is kept here. So the hormones are made and released from the endocrine glands, and via blood circulation they reach their target cells via blood circulation, having receptors against these hormones. And here you see the neuron, an electrical signal arrives in the synaptic area and it causes the release of neurotransmitters such as acetylcholine, adrenaline, nitric oxide, gamma aminobutyric acid, etc. So it can either depolarize or hyperpolarize the opposite cell. By depolarization, it will stimulate the neighboring cell, or by hyperpolarization, it will inhibit it. If it is not a neuron, if it is a muscle cell, then it can cause contraction in the muscle. We see this in neuromuscular junction. Okay. So the paracrine and autocrine communications are also shown here. We will see in the text in coming slides, but shortly, paracrine communication is that the cells release chemicals to the extracellular fluid, and these chemicals will affect the cells nearby. So this is called paracrine communication. And autocrine communication is that the substances released by the cell to the extracellular fluid will affect the cell itself. So it is called autocrine communication. So a hormone is a chemical messenger that enables the hormone secreting cell to communicate with other cells, with the blood acting as the delivery system. The cells on which hormones act are called the hormone's target cells. Hormones are produced in and secreted from the endocrine glands or in scattered cells that are distributed throughout an organ. They have important functions in essentially all physiological processes, including growth, reproduction, metabolism, mineral balance, and blood pressure, and several of them are produced whenever homeostasis is threatened. In contrast to hormones, neurotransmitters are chemical messengers that are released from the endings of neurons onto other neurons, muscle cells, or gland cells. A neurotransmitter diffuses through the extracellular fluid, separating the neuron and its target cell. It's not released into the blood like a hormone. In the context of homeostasis, they form the signaling basis of many reflexes, as well as having a vital role in compensatory responses to a wide variety of challenges, such as the requirement for increased heart and lung function during exercise. Chemical messengers participate not only in reflexes, but also in local responses. Chemical messengers involved in local communication between cells are known as paracrine substances or agents. Paracrine substances are synthesized by cells and released once given the appropriate stimulus into the extracellular fluid. They then diffuse to neighboring cells, some of which are their target cells. Given this broad definition, neurotransmitters could be classified as a subgroup of paracrine substances, but by convention they are not. Once they have performed their functions, paracrine substances are generally inactivated by locally existing enzymes and therefore do not enter the bloodstream in large quantities. Paracrine substances are produced throughout the body. An example of their key role in homeostasis is their ability to fine-tune the amount of acid produced by cells of the stomach in response to eating food. There is one category of local chemical messengers that are not intercellular messengers, that is, they do not communicate between cells. Rather, the chemical is secreted by a cell into the extracellular fluid and then acts upon the very cell that secreted it. Such messengers are called autocrine substances or agents. Frequently, a messenger may serve both paracrine and an autocrine functions simultaneously. That is, molecules of the messenger released by a cell may act locally on adjacent cells as well as on the same cell that released the messenger. This type of signaling is commonly found in cells of the immune system. So on the right hand side you see the paracrine and autocrine communications. A point of great importance must be emphasized here to avoid later confusion. A neuron, endocrine gland cell, and other cell type may all secrete the same chemical messenger. In some cases, a particular messenger may sometimes function as a neurotransmitter, a hormone, or a paracrine or autocrine substance. Norepinephrine, for example, is not only a neurotransmitter in the brain, it is also produced as a hormone by cells of the adrenal glands. All types of intercellular communication described thus far in this section involve secretion of a chemical messenger into the extracellular fluid. However, there are two important types of chemical communication between cells that do not require such secretion. The first type occurs via gap junctions, which are physical linkages connecting the cytosol between two cells. Molecules can move directly from one cell to an adjacent cell through gap junctions without entering the extracellular fluid. In the second type, the chemical messenger is not actually released from the cell producing it, but rather is located in the plasma membrane of that cell. For example, the messenger may be a plasma membrane protein with part of its structure extending into the extracellular space. When the cell encounters another cell type capable of responding to the message, the two cells link up via the membrane-bound protein. This type of signaling, sometimes termed eustacrine, is of particular importance in the growth and differentiation of tissues as well as in the functioning of cells that protect the body against pathogens. It is one way in which similar types of cells recognize each other and form tissues. Thank you very much for your attention.